Hi, I hope you're doing well in these strange days. I'm here for this kickoff of our project, the Nelore hologenome implications in beef quality and feed efficiency. I'm Luciana Correia de Almeida Regitano, the coordinator of this project, and I'm speaking on behalf of Ademar, Gerson, and Luis, which are the principal investigators. So in this slide, we have the presentation outline, which is supposed to be the project overview, goals, specific goals with the progress up to now linked to each goal, activities schedule, and approved budget. For the project overview, let's remind that we have biological samples and data from two different experiments. One is the beef quality, which was coordinated by myself and it was financially supported by Embrapa. And the other is the water footprint project that was coordinated by Dr. Júlio Palhares and was financially supported by the National Council of Science and Technology in Brazil, CNPq. From the beef quality project, we had 34 sires that were made to commercial dams, and we produced 800 steers across 3 years in 5 different farms. And we had phenotypes scored for growth, beef production and quality, feed efficiency, mainly residual feed intake. From these animals, we also had genotype on high-density SNP sheep. And when the project was finished with Embrapa support, we got an extra. Well, when this Embrapa's project was finished, we applied for a FAPESP budget, and with these we were able to sequence the genome of 20 sires, the ones that had the higher progeny size that had residual feed intake data recorded. We also did the RNA, mRNA and miRNA sequencing from approximately 190 muscle samples and 30 liver samples, and we did the proteomic profile of 65 and 40 muscle samples. These were done in 2 different equipment, so we have to consider them as separate data. And here is just an illustration of what we did with this data. We did many GWAS analysis for different phenotypes, and we also did gene expression differences between extreme phenotypes, both for liver and for muscle, but we used this approach in several different. We also described the copy number variation in this SNP data, and we associated this copy number with gene expression. So this is just a sample. It's in the first, altogether, the first thematic project yielded 28 different papers. From the water footprint project, we had 2 groups of bowls with 26 animals each group, and one of the groups received regular diet and the other byproducts diet. We collected from this animal growth, beef production, feed efficiency, as we did in the first project, but we also collected methane emission and water footprint in these animals. We collected feces and ruminal content. Feces were collected in the end of the feedlot. Ruminal content, ruminal epithelium, liver, and muscle were collected in the slaughterhouse. We then approved a CNPq project, Applied Metaomics and Brazilian Bos Indicus, in which we were supposed to sequence the metagenome of feces and ruminal content, obtain the metabolites profile and the mineral content profile of these samples. But there was a shortage on the budget since only partial budget was liberated, and we had to exchange whole metagenomic sequencing by 1618S sequencing. Now, with this newly approved FAPESP Nelore hologenome project, we wish to obtain metabolites, mineral content profile, RNA-seq, miRNA-seq, and proteomics from ruminal epithelium, liver, and muscle. And we also want to obtain the lipidomics profile of muscle. It is also a decision we have to take if we want to use the overhead for metagenome sequencing and obtain much profound information on this metagenome. So there are three general goals in this project. The first is to study the interaction of host microbiome genomes and its relation to feed efficiency, lipid profile, methane emission, and indirectly to water footprint. The second is to explore data from a previous thematic project, expanding the search for regulatory mutations that explain the main differences in mRNA abundance related to beef quality and feed efficiency traits in Nelore beef cattle. And the third is to create a set of data loading tools capable of handling the results of the analysis executed in this project, as well as an application programming interface and web interfaces to query the data. So finally, the relational data bank. The specific goals, now I'm going to tell you what's being done or what we have to do to approach of the specific goals in this project. The first specific goal is to explore the taxonomic diversity and functionality of the rumen microbiome of Nelore bulls submitted to two different diets using metatranscriptomics, associating this diversity with diet, methane emission, and production traits. So to access the taxonomic diversity of the GIT microbiome, we published this paper, which is headed by Bruno Andrade, in which we looked at the interdependency of the two environments, rumen and column. And what Bruno found in this study was from 4,200 approximately amplicon sequence variants, he found 96 bacteria and 47 archaea, which were common between the two environments. And the shared archaeal ASVs between microbiomes suggested that there is a dependency of the predominant fecal methanogen population on the rumen population. So it's kind of the methanogenic population and the feces are not exactly a result of colonization in the colon, but it's much more a carryover from the rumen to the feces. Well, the second paper that's being prepared right now, it's in the hands of the co-authors, is a paper on phenotype microbiome association, and it's been done in collaboration with Haytham Affley from the Cork Institute for Technology in Ireland. And in this manuscript, what Bruno describes is, like here in this graphic, you can see bacteria and archaea and feces, and the same as bacteria and archaeas and the rumen. And you can see there are both positively related bacteria with methane emission and negatively related to methane emission. The same happens with archaea in feces. So he found 12 bacterial ASVs associated to methane emission, 5 being positive and 7 negative. He also found 5 archaeo ASVs associated to methane emission, being 3 positive and 2 negative. So these are very interesting results because one of the problems that we had in hands was to prove that feces are a good indicator, can be used as a good indicator for methane emission. And here what he saw is that he found more ASVs associated to methane emission in feces than he found in rumen. So this is awesome. So for the other activities on these specific goals, we have approved a postdoc position for 24 months. We still need to describe the activities and to announce and hire this postdoc. But at this moment, facing all the problems related to COVID-19, economic crisis, and currency exchange, we have to wait a little bit so we can have the proper conditions for this professional to execute his work. And there are some technical challenges also regarding this activity because we wish to sequence both coding and non-coding RNAs, so there will be no poly-A selection. And so we are at this moment thinking about which kits to use. There are some options from Illumina, but if anyone has tips on ribosomal RNA depletion methods, they are really welcome. The second specific goal of this project is to explore the metabolic diversity of the rumen microbiome of Nelore bulls by means of metametabolomics, associating this diversity with diet, methane emission, and production traits. And for this, we have rumen, feces, and muscle metabolites profiles done. Here is preliminary results from the metabolites. So you can see here the ruminal fluid of Nelore, how it resembles. It's a quite complex metabolite profile. And this is Jessica's Malheiros postdoc project. She is working on the quantification of differentially expressed metabolites in ruminal fluid, feces, and muscle. And she is at this moment writing her first manuscript. So beside the polar metabolites, she also obtained non-polar metabolites. And the next steps for the metabolome is to obtain liver metabolites and to verify these profiles of liver metabolites by 2D NMR and to publish this information. So the third specific goal is to explore the functional genomic profile in the rumen epithelial tissue and muscle, liver through transcriptomics, proteomics, metabolomics, and lipidomics of Nelore bulls submitted to different diets, associating to feed efficiency, carcass, and beef quality, including mineral content. And we have also RNA extraction done for ruminal epithelium samples. Few samples had a bad integrity, but we already re-extracted them. The spectrometry was great. Let's check if the integrity of these RNA samples are also okay. But at this moment, all lab activities are waiting for a better economic scenario, especially for exchange rates because we need a lot of foreign consumables for these activities. So unless it's very important for a student project or for something that cannot be held, we are holding down these activities. As the fourth goal, we have to identify and annotate mRNAs in the ruminal content, rumen epithelial tissue, and muscle. And we have already, as I told you, a total RNA extraction from this epithelial tissue. And we are organizing the activities to start sequencing the mRNA. This data on mRNA sequencing is going to be part of the master's degree project of Tani Porto. Other data processing regarding the rumen epithelial tissue will be assessed by the postdoc to be hired. The fifth specific goal is to characterize lipid profile and inflammatory lipid mediators in skeletal muscle and the relationship with tag deposition. This was a postdoc position that was included in the primary budget when we submitted the project to FAPESP, but it was cut down from the budget. At this moment, we submitted a new postdoc proposal to FAPESP in name of Gelson Vieira, and this is being done in collaboration with William Festuccia. We also wish to characterize protein and mineral content profiles in rumen to associate it to diet, microbiome, feed efficiency, carcass, and beef quality. And the mineral profiles have been obtained already by Anahita, a colleague from Embrapa, and her postdocs and students. And she's also dealing with the proteomics. She started some tests with muscle proteomics, and she's getting good sample preparation, but there is a little trouble regarding ruminal content. The samples from ruminal content are very, very fuzzy. We need some, if anyone has experience on ruminal proteomics, please give us a note. Another goal of this project is to explore the quantitative effects of transcriptome-wide mRNA levels on traits. And this is quite different from what we've done in the previous project, where we're all the time or most of the time comparing individuals representing extremes of one phenotype. In this case here, we want to have an idea of the complete distribution of mRNA, of gene expression in the population, and to have something like an additive effect related to each gene expression. So this is one of the works that, the papers that is related to this goal. It was headed by Andressa Lima in collaboration with James Coates. She was an internship in his lab for 6 months. And in this paper, they describe the transcriptome-wide mRNA level effects on feed efficiency. And they showed that the expression of these guys here is associated with multiple feed efficiency related traits in a continuous form. So it's not only when you compare extreme animals that you find this effect. You can find it as an additive effect. Another published paper that relates to this objective is this paper by Wellison Diniz in collaboration with Haja Kadarmidin, and it deals with the co-expressed pathways of a continuous sample of mRNA RNA-seq. So he worked on the RNA-seq data of 194 Nelore steers, and he used this information to associate to 3 meat quality traits and the concentration of 13 minerals. So by doing this, he was able to find evidences of the interplay among gene expression, mineral concentration, and meat quality traits. And there is another article, another published paper related to this specific goal, which deals with expression levels by qPCR of specific genes that were related in our differentially expressed analysis. And in this work, Wellison described qPCR liver expression profiles in 83 animals, and he analyzed 6 target genes, from which he found positive regression of FABP1 gene expression related to residual feed intake and dry matter intake. And he also found a positive regression effect on this solute carrier gene expression with Kleiber index and residual growth rate. We are also committed to estimate the effect of different SNP subsets based on their function and the accuracy of genetic prediction. Related to this goal, there is a work in progress which is dealing with inferring the effects of eQTL variants in genomic prediction models. This work is headed by Aline Cesar and Juliana Petrini. And sure, this is one initiative, but there is room for many more SNP prioritization approaches. So we can think about using ASE QTLs, SNPs in QTLs, and SNPs in miRNAs, and a number of different features associated to these SNPs. And most of what's been done until now is being done with Illumina chip SNPs, but we do have genome-imputed SNPs. Another specific goal is to integrate multi-omic data SNP, genotypes, mRNA, miRNA, and proteomics to explain the relationship between these elements, and also to expand the description of regulatory genomic elements and genes associated to phenotypes. Some integration was done in the previous thematic project. We did eQTL analysis, eCNV, miRNA, mRNA networks. We also did miRNA protein networks. And additionally, a number of articles was published between the formal end of the first and the approval of the second FAPESP project. So these here are some examples of the results we have achieved related to this goal, like the integrative analysis of microRNAs and mRNAs, relating to composition and metabolism. There is also this work by Mirelle Poletti, where she describes the proteomics, the label-free quantitative proteomics of muscle and its relation with intramuscular fat deposition. In this paper, Mirelle describes the integration of mRNA and proteomic data, and she found 17 differentially expressed genes corresponding to differentially abundant proteins. 3 genes and proteins did not agree on the direction of the fold change between the groups, and when she looked at the microRNA data, she was able to explain 2 of 3 unexpected mRNA protein relationships. Still regarding this specific goal, there is this fresh from the oven publication where Mirelli also describes the proteome alterations associated with oleic acid and cis9 trans 11 conjugated linoleic acid in bovine muscle. And in here, she describes the comparison of 10 high and 10 low animals for each fatty acid. And she found 103 differentially abundant proteins for oleic acid and 133 for C9T11 CLA. And considering proteomic versus transcriptomic, she found 8 correspondences for oleic acid and 10 correspondences for C9T11. And this work here is a result of an internship of Priscila Oliveira in Jim Reese's lab. And here they deal with the co-expression networks related to regulatory roles of mRNAs in fatty acid composition. And using this approach, they were able to describe a number of mRNAs and genes that are potential regulators of fatty acid synthesis in muscle of Nelore. And this work here, which I mentioned to you regarding quantitative effects of gene expression, but it also deals with many integrative analysis for mineral concentration, meat quality, and gene and microRNA expression. And by the time this project was approved, we had 3 articles submitted to publication. The first of these 3 papers is published since 21st May, so a few weeks ago. And it was a work by Juliana Afonso in collaboration with Marina Fortes and Antonio Reverter, in which they describe the use of a new co-expression and regulatory impact factor approach to describe regulators of mineral amount in Nelore cattle muscle. And so what's new in this approach? They included the GEBVs as additional nodes in the gene network. So they had both the gene expression, the microRNAs, the mRNAs, the microRNAs expression, along with the GEBVs, the continuous variation of GEBVs for these animals. So this is a detail of figure 3, where we have the co-expression networks among genes, mRNAs being part of enriched pathways, which are either differentially expressed or correlated to a mineral, or both. And we have different elements. We have hub genes, transcription factors, mRNAs, or presenting a significant reef regarding 9 of the minerals in study. In this case, I'm showing you the magnesium-related regulatory elements. And then you can see here, for example, this is a microRNA, which is the symbol here, microRNA, and it has a significant reef because it's orange. And here you have hub gene, and as it's green, it's down-regulated, and as it has a star on top of it, it's a correlated and also a differentially expressed gene. So this is a quite awesome paper. It has many information on how these elements cooperate to regulate the concentration or the amount of mineral in the muscle. The second manuscript that was submitted by the time this project was approved is about the allele-specific expression in Bos indicus muscle. And this manuscript, in fact, was a result of the previous thematic project. And we submitted it to publication in scientific reports and got a feedback from the editor saying that it would be much better if we used the new version of the genome. So we decided starting over again and do all this analysis with the new bovine version. And in this paper, we integrate the SNPs in the high-density bovine, Illumina bovine sheep with muscle RNA-seq, and after filtering, because we can only test heterozygote SNPs, and you also need a good number of reads from each of these loci. So at the end, we were only able to test about 1,000 SNPs, and from these, Marcela showed that 820 had an imbalance between the expression of the two alleles. And these are good indicators that nearby we have a regulatory mutation. This manuscript was accepted and at the end of May, so it's in press right now. And the third manuscript is a consequence of this previous allele-specific expression, and it describes the ASE-QTL, or the SNPs that might explain the difference between the expression of the two alleles in Marcela's work. And this was a master's degree project from Jennifer Bruscadin. And I can show you here some of the results in numbers. She studied the 820 ASE SNPs, and from this she was able to find ASE-QTLs for 126 ASE SNPs. These were regulated by 1,134 ASE-QTLs, from which 360 had evidence of regulatory function and 404 were overlapped with QTLs. The ASE-QTLs with regulatory indicatives, from these, she found 215 within transcription factor binding sites, 162 were within miRNA binding sites, and 16 were methylated ASE-QTLs. And from the ASE-QTLs overlapping QTLs, 234 overlapped Nelore QTLs from our project, and 173 overlapped QTLs from QTL database. And so here is a figure of the distribution of these ASE SNPs and ASE-QTLs in each result section. So we have here in gray the ASE SNPs and this layer here. The red layer are ASE-QTLs that regulate these ASE SNPs. And we have in blue QTLs and this layer here. And we have transcription factor binding sites in this section here. And we have miRNAs in green. And we have methylation in purple, like this one. And we can see some examples where we have a good overlap of a lot of different features. As expected, most of the ASE-QTLs were in fact big blocks of LD, and here is one example of a very interesting block, which is the biggest ASE-QTL LD block, which contained 21 ASE-QTLs and was associated with the allelic expression of CMYA5. All the SNPs in this block were within miRNA sites, so this block here is all within miRNA binding sites. And this blue star represents a tag SNP here, and the position in the genome of this tag SNP is this red dotted line. And you can see here in this figure that you have 2 isoforms of this gene and a third isoform. So this tag SNP is shared between the 2 top isoforms and not by the bottom isoform. It is within the intronic region of these isoforms, and it lies within a transcription factor binding sites. The ASE-QTLs in this block overlap 7 meat quality traits, including back fat, light color of fat, ribeye area, Warner-Brotler shear force in 2 different times, and water holding capacity. Still related to this specific goal, there is another work in progress, which is the PhD project of Francisco and Coutinho's lab. So Francisco is using a Bayesian approach to build networks combining GBV of 109 traits in 386 animals. This is being done in a partnership with Gota Muruta's lab, and this is how this network resembles. I'm pretty sure if you're interested, Francisco can tell you more about that. So still from Francisco's work, you can see here in this network that you have multi-omic data integration, you have phenotypes, you have miRNAs, and then you have models of gene expression, you have proteins, and so on. Still related to this main specific goal regarding expanding the description of regulatory genomic elements, we have the work by Tainá Cardoso, who's a postdoc sponsored by FAPESP. She's dealing with variations in regulatory regions associated with meat production and quality characteristics in the Nelore breed. She's mainly focusing on miRNAs. And there is the work by the undergrad Karina Santos, who's working on the gene regulation mechanisms related to feed efficiency in Nelore cattle. And as a result, related to this description of regulatory genomic regions, we also have results in the published manuscript by Andressa Lima in collaboration with James Coates, also describes some regulatory elements. So speaking about the project of Karina Santos de Oliveira, she got a bunch of genes that in previous studies of the first thematic project we found as differentially expressed for groups of animals contrasting regarding feed efficiency. And there was also this gene KCNJ11 whose expression was related to meat tenderness and feed efficiency, and it lies within a QTL region and muscle. for QTL region for Warner-Brotzer shear force. So the aim of this project is to identify functional DNA variants of these genes and describe their likely regulatory impact for annotation and to verify their association with phenotypic measures. And as you see, we have quite a number of results related to this specific goal, and there are still other activities that are ongoing at this moment, like Mirelle Poletti is working on the proteomics of Warner-Brotzer shear force extremes. She's also working on the general proteomics, considering all the samples we have proteomics, and integrating this information to mRNA and miRNA, regardless of phenotype. And she also is dealing with a double G-CNA approach for protein versus phenotypes. Well, and if you think this is not enough, there are more ongoing activities regarding this specific goal. There is a pQTL analysis being conducted by Mirelle Poletti, and she also has the profile of muscle metabolomics of a subsample of 200 steers that have RNA-seq data. And she's working on this data for a manuscript at this moment. And there is also the splice QTL analysis that's being conducted by Poliana Cisiotto. There are some challenges that I would say it's not exactly a challenge, but we have a lot of work ahead because all the data on QTL, eQTL, CNV, differentially expressed genes, differentially expressed miRNAs, and so on, are in UMD 3.1. So for all this integration analysis, we should put them all in the new version of the genome. There are perspectives for this reanalysis for CNV, which is a postdoc project by Vinicius Silva in Coutinho's lab. It's being analyzed by FAPESP at this moment to see if they're giving us this position for the CNV analysis in the new version of the genome. And Aline Cesar is working on the eQTL analysis for the new genome version. And the other point that we have to consider is that most of what we did, I think that beside the work by Karina Oliveira and Tainá Cardoso, all the others are based on Illumina SHIP SNPs. But we have genome sequenced, genome level imputed SNPs. We have SNPs from RNA, and we have miRNA sequences also to retrieve SNPs. This SNP call is done, the imputation from genome level is done, and we have all the SNP calls from these two sequencing here, but we should perhaps reanalyze GWAS in the new version of the genome, but with imputed SNPs. And finally, the 10th specific goal is to extend the development of the software Machado by creating a set of data loading tools. This is in the direction of constructing a database which will enable us to query this data. This activity is being headed by Ademar with his team at Embrapa Informatics, and we count on the cooperation of Chris Elsik for this. In fact, it's not that it's not started, but we 2 weeks ago had a meeting and decided to start having a look at this data, organizing the information, how each data resembles, how this information is in files and so on. So we can start like dating this information and have a plan to start with. For this activity, we have in the project budget a position for a bachelor in TI for 24 months and in full dedication. And we are waiting for the proper opportunity to implement, to select this professional. Well, I hope I gave you a good idea of the project. What do we have to do? What's done? What is being done? And what do we expect from these activities? I'm not going to go through all these activities schedule because it's too much. It's 5 years, very detailed in any of the procedures that we have to take to achieve our goals. But you will see that in the activity schedule, I named some persons, I listed some people, but I wish you have the time to look at this schedule or to this general information and tell me, okay, I would like to give a hand on this, or I would like to be involved on that, regardless of having names listed in these activities. So this is not a closed or defined situation. Please feel free to tell that you wish to be involved in any of these activities. So let's take the example here of the first year of the project. The project starts at December 19, so we are in the 7th month of the project, and here I describe each activity, very detailed activity like total RNA extraction, and I told you where we are in relation to this timetable. So you see here, we were supposed to finish this total RNA extraction from all the tissues, but we just have ruminal epithelium extracted because we are facing a different situation. I wouldn't be worried about this because although we are kind of making a big frame shift in this timetable, we have plenty of results and we have a good explanation for our delay. Like here, everything here is done already. And so this is how the timetable resembles. And in this column here, you have a brief description or a tentative description of the research team. So you have these activities here as part of postdoc position, but you also have Tainan, Priscila, Karina, and Wellison that were working on this activity, and so on. This is going to be for the 5 years of the project, this kind of information. And here there are more activities for year 1 of the project, like DNA extraction and genotyping for the 52 animals is done already, but we still need to do the imputation. And we have also metabolic profile, which is done. SNPs imputation for the first project data set is done too. There are people working on the genomic predictions. So as you see, next years are much more lighter in terms of activity because the first year was the concentration of sample preparation and profile obtention. And more activities for the second year of the project, but we know, like here, the activities on AS-EQTL are done, and so on. So the timetable is a lot shifted from what it was proposed initially. We have here people working already in genomic prediction, and SNPs and mRNA sequences is almost done too. So on for and the 5th year of the project. In this slide, I show you the approved budget. We had only one cut down from what we originally proposed, which was that postdoc position for lipidomic. So we approved exactly the amount we asked for. But there is a concern because Brazil is facing a big economic crisis at this moment. And this is impacting a lot our currency. So we are experiencing devalorization of our currency in face of US dollar. And this is the description of the scholarships or the positions we approved for this project. We asked for 2 postdoc positions, but only one was approved for 24 months. And we also approved 1 bachelor in informatics in TI for also 24 months. So I hope this will be a great opportunity for us for doing an excellent job as we did in the first project. And I wish we can strengthen old collaborations and start very nice new ones. And with this, I thank you so much for your attention and wish that we get closer and be able to meet each other pretty soon. Thank you so much. Please, any comments sent to my email. Thank you.